Good evening. My name is Jui Pandhare, and I'm an assistant professor in Meharry Medical College in Nashville, Tennessee. And today I'm going to talk about our ongoing work on the neuropsychiatric effects of HIV-1 integrase inhibitors. So we all know that HIV is a global pandemic. And here in the United States, HIV-1 has now become a major health disparity. This is because although African Americans are just 13% of the US population, more than 42% of new infections are diagnosed in African Americans. Now, even though HIV is a problem, because of the availability of the anti-retroviral drugs, HIV-1 has become a manageable disease. And there are several HIV drugs that target the various steps of the HIV life cycle. These include the reverse transcriptase inhibitors, protease inhibitors, as well as integrase inhibitors. And in recent times, integrase inhibitors are the frontline drugs. They are the most prescribed drugs in therapy, and because they very effectively control the virus and they have a very high barrier to resistance. And there are 5 integrase inhibitors or INSTIs, integrase transfer inhibitors, that are currently used in therapies. However, some of these inhibitors, specifically dolutegravir, has been associated with adverse neuropsychiatric effects. And the underlying causes that are responsible for these neuropsychiatric effects are not known. And so therefore, the goal of our study is to understand the mechanisms that are involved in these adverse neuropsychiatric events. So to begin this study, we used a very minimalistic model of differentiated neurons. We treated them with 2 integrase inhibitors. One is raltegravir and the other is dolutegravir or DTG. And we just observed what happens to the neurons. And interestingly, what we saw that in the control cells, as well as the raltegravir treated cells, the neurites were not long. You can see the length were nice and long, while when the cells were treated with DTG, there was a marked reduction in the neurite length, suggesting that there might be some alterations in the neuronal communication. So to further understand, we carried out RNA sequencing analysis to identify some of the genes by knowing the global neuronal transcriptome. And this also, we observed that DTG markedly upregulated genes were very different from those that were affected by either raltegravir or control. And when we further analyzed these genes, what we found that specifically genes were dysregulated in glutamate and calcium signaling that have been implicated in neuropsychiatric disorders. So we focused on particularly 2 genes. One is the GRIN2D gene, which codes for the NMDA2D subunit of the NMDA receptor. And the other ones are the low voltage gated calcium channel genes, is shown over here, CACNA1G, H, and I. And so we selected these genes and we validated them again in our differentiated neuronal model, as well as we collaborated with Vanderbilt University to further validate in a mouse model where we treated mice with these integrase inhibitors, and from the hippocampus, we again performed RNA studies to see if the same genes are affected, and we indeed found that the same genes were also affected in mice. And so now in future, what we have proposed a hypothetical model where we want to check whether the INSTI-mediated dysregulation of glutamate and calcium signaling within the brain reward circuitry is affecting the synaptic communication that can lead to neurodegeneration. So here what we propose is that within a synapse, because of the stimulation of low voltage gated calcium channels that will affect the release of glutamate, stimulating the NMDA2D subunit, causing excitotoxicity and leading to degeneration, that might lead to these neuropsychiatric effects. So from our studies, we hope to identify novel pathways that may be targeted to reduce these INSTI-associated adverse neuropsychiatric effects. And that is the work that is currently going on in the lab using electrophysiological studies to understand these effects. So thank you very much for listening to my presentation, and I look forward to questions. Thank you.